Hello students, welcome to EPG Pathshala program. I am Dr. Shoaib Lukman from the Council of Scientific and Industrial Research, Central Institute of Medicinal and Aromatic Plants, Lucknow. Today we are going to talk on module Lipids, Inborn Errors of Metabolism, Part 4 from the paper Lipid Metabolism. The objectives that I am going to cover in this module is to understand the inborn errors of metabolism associated with the peroxisomes. What are the disorders and diseases linked with the peroxisomal dysfunctions, and how the beta as well as omega oxidation disorder takes place. If we look at the peroxisomal disorders, they are of three types. The first, the peroxisome biogenesis disorder or the PBDs. The second, the single peroxisomal enzyme protein deficiency, and the third, the single peroxisomal substrate transport deficiencies. The peroxisomal disorders represent defects in the peroxisomal functions leading to genetic diseases in human. The peroxisomal enzyme deficit group comprises 7 different disorders, of which phytanoyl coenzyme A hydroxylase, which is responsible for adult Refsum disease, and D bifunctional protein deficiencies are the most recurrent. The X-linked adrenoleukodystrophy is the only disease of the peroxisomal substrate transport deficiency group, and the therapeutic interventions are typically restricted to an X-linked adrenoleukodystrophy. The most frequent adult onset peroxisomal disorders are X-linked adrenoleukodystrophy and Refsum disease, recognized biochemically by an elevated phytanate concentration and a very long chain fatty acid respectively. Nonetheless, only 85% of female carriers of adrenoleukodystrophy have unusual plasma concentrations, and it ought to be assumed in mind that distinctly unlike phenotypes of adrenoleukodystrophy can arise in one family. 2-methyl coenzyme A racemase deficiency presents an instance of a lately described adult onset inborn errors of metabolism. The clinical features of this deficiency are analogous to the Refsum disease and typify biochemically by the elevated concentrations of plasma pristinate. Quantifiable features that include peripheral neuropathy, pigmentary retinopathy, and seizures. Acute and subacute appearance crop up by fever, pregnancy, and rapid weight loss. In this pictorial presentation, you can see that the PBD group comprises 4 different disorders. The first is neonatal adrenoleukodystrophy, which is called as NALD. The second is Zellweger syndrome, ZS. The third one is the infantile Refsum disease, the IRD. And the fourth is rhizomelic chondrodysplasia punctata, RCDP. Cerebrohepatorenal syndrome, which is also known as Zellweger syndrome, is a congenital and rare disorder, illustrated by the absence or reduction of peroxisomes in the cells of the brain, kidneys, and liver that distress the aptitude to beta-oxidize very long chain fatty acids in the peroxisomes of the cell owing to a genetic disorder in one of the numerous genes involved with peroxisome biogenesis. A number of peroxins are interlinked with Zellweger syndrome, including PEX1, PEX2, PEX3, PEX5, PEX6, PEX12, PEX14, and PEX26. This cerebrohepatorenal syndrome disorder is also known as the Zellweger spectrum occurs in one of the three peroxisome biogenesis disorders. The further two diseases are infantile Refsum disease and neonatal adrenoleukodystrophy (NALD). The defects associated with the beta-oxidation of very long-chain fatty acid. A very long-chain fatty acid in which carbon atoms are 22 or more than 22 that undergoes beta-oxidation in the peroxisomes, and the short-chain fatty acids that undergoes beta-oxidation in the mitochondria. There is a crosstalk between the peroxisomes and the mitochondria through octanoyl-CoA or octanoyl-carnitine happens in the cell during the lipid metabolism as depicted in the figure. In contrast to the mitochondrial beta-oxidation, the preliminary dehydrogenation in peroxisomes is catalyzed by an FAD containing acyl-CoA oxidase. The FADH2 produced is oxidized by molecular oxygen, which is abrased to hydrogen peroxide. Thus, as a result of which, no adenosine triphosphate is generated by this step. The hydrogen peroxide is reduced to water by catalase. There is a note that genetic defects either in the ability to target matrix proteins to peroxisomes, resulting in the Zellweger syndrome, which is a peroxisomal biogenesis disorder, or in the ability to transport very long chain fatty acid across the peroxisomal membrane, resulting in X-linked adrenoleukodystrophy that lead to the accumulation of very long chain fatty acid in the blood and tissues. Let us talk about the disorders associated with the omega oxidation of fatty acids. As we all know that phytanic acid, which is a branch chain 20 carbon fatty acid, undergoes omega oxidation. This fatty acid is not a substrate for acyl-CoA dehydrogenase because of the presence of the methyl group on its beta carbon. Instead, it is hydroxylated at the alpha carbon by phytanoyl coenzyme A alpha hydroxylase. Carbon 1 is released as carbon dioxide and the product 19 carbon pristanic acid is activated to its coenzyme A derivative and undergoes beta oxidation. Refsum disease is one of the example of this kind of disorder, which is an autosomal and rare recessive disorder caused by the absence of peroxisomal phytanic hydroxylase. This results in the accretion of phytanic acid in the tissues and plasma. As a result of the accumulation, it depicts symptom like neurologic, and the treatment involves the dietary restriction to halt the disease progression. There is one point to be noted here, that omega oxidation at the methyl terminus is also known and generate dicarboxylic acid. Normally, a minor pathway of the endoplasmic reticulum, its upregulation is seen with conditions such as MCAD deficiency that limit the fatty acid beta oxidation. The mechanism of the Refsum disease: the phytanic acid oxidizes to yield carbon dioxide and pristanic acid, and this pristanic acid is an odd chain fatty acid. This oxidation process and the conversion is carried out by phytanic acid alpha oxidase. The so formed pristanic acid is consequently oxidized by beta oxidation. This process rivets hydroxylation of the alpha carbon removal of the terminal carboxyl group, and concomitant alteration of the alpha hydroxyl group to a terminal carboxyl group, and the linkage of coenzyme A to the terminal carboxyl group. This branch substrate will operate in the beta oxidation procedure, eventually yielding acetyl coenzyme A, propionyl coenzyme A, and 2-methylpropionyl coenzyme A, whereas isobutyryl coenzyme A in the case of phytanic acid. The pictorial representation in the slide shows the Refsum disease of an adult with defective gene PEX7 and PHYH. Due to this defect, there is a mutated ocular cells as phytanoyl-CoA cannot be further converted. Due to the lack of phytanoyl-CoA hydroxylase activity, hence accumulation of phytanic acid and other compounds takes place which are toxic to the cells, resulting in the clinical symptoms. In a smaller percentage of cases, PEX7 is mutated so that import of the enzyme into the peroxisome is defective. The inborn errors of cholesterol biosynthesis. Cholesterol have an important function as it is a part of membrane component as well as it is a precursor to bile acid, vitamin D and steroid hormones. The cholesterol biosynthesis disorders have lately emerged as an imperative inborn errors of metabolism, which have taught us a lot of new genetic and biochemical edifications, despite the fact that most of the metabolic diseases are exemplified by exclusively or largely postnatal biochemical deficiencies or toxicities. Cholesterol biosynthesis disorders are prominent for their relentless effects on prenatal development. The pictorial representation of cholesterol shows the number of carbon atoms in the cyclopentanophoranthrene ring, as well as the side chain and methyl groups and the site of esterification. The synthesis of cholesterol from lanosterol and the syndromes and inborn errors of metabolism associated with them. There are numerous disorders that are associated with the synthesis of the cholesterol, starting from the lanosterol and its conversion to dihydroerythrosterol. Due to lack of lanosterol 14-alpha methylase, a disorder named as Antley-Bixler happens. Due to the deficiency of sterol 14 reductase or the lamin B receptors, HEM dysplasia happens. A deficiency in C4 demethylation complex, child SC4 mole disorder happens. Defect in sterol isomerase causes CDPX2 Lithosterol 5-desaturase deficiency causes lithosterolysis. 7-dehydrocholesterol reductase deficiency causes Smith-Lemli-Opitz syndrome, and sterol 24 reductase causes desmosterolosis. One of the very peculiar type of the cholesterol inborn error is familial hypercholesterolemia. The familial hypercholesterolemia, also known as the spelled familial hypercholesterolemia, FH or SFH, is a genetic disorder portrayed by the high cholesterol levels, exclusively very high levels of LDL, low density lipoprotein or bad cholesterol, in the blood, and ultimately cardiovascular illness. Individuals with FH or SFH display different body biochemistry, and their elevated cholesterol levels are less responsive to the control methods of cholesterol, frequently more efficient in people without FH or SFH. For example, the dietary modification and statin tablets. However, the high doses of statin as a treatment regimen is usually effective. Smith-Lemli-Opitz syndrome. The Smith-Lemli-Opitz syndrome, SLOS, is a manifold congenital anomaly, which is also abbreviated as MCA, or the mental retardation syndrome, caused by a deficiency in the synthesis of the cholesterol. So, Smith-Lemli-Opitz syndrome is an autosomal recessive genetic state caused by the deficiency of 3-beta hydroxysterol delta 7 reductase, or DHCR7, with an EC classification of 13121. This is the ultimate enzyme in the sterol biosynthetic pathway that convert 7-dehydrocholesterol to cholesterol. The other lipid disorders include carotenemia. As its names indicate, that it is a condition where carotene is found in blood. Almost 111 years back in 1904, it was originally noted as xanthosis diabetic by Van Noorden, who experiential it to be well known in the nasolabial folds and on the palms and the soles. Carotenemia is a clinical condition depicted by the yellow pigmentation of the skin, that is xanthoderma, and this condition is augmented by the presence of high beta carotene level in the blood. In most of the cases, the situation pursues the extended and too much consumption of carotene-rich foods. That is, example are carrots, sweet potatoes, and squash. Also, Carotenemia is a frequent finding in children. The condition of carotenemia is undisruptive and it can show the way to a mistaken diagnosis of jaundice. If we see that carotene is a lipochrome that usually append yellow color to the skin, and with the elevated blood level of carotene, the eminence of this yellowing is amplified. When the stratum corneum is thickened, or when the subcutaneous fat is strongly represented, carotenemia is particularly evident. The situation is more effortlessly appreciated in light complex people and can be well monitored by the yellowing of the palms and the soles in the darkly pigmented individuals. The causes of the carotenemia. In infants and children, the diet-induced carotenemia has been observed most frequently. Mothers might tempt the firm by offering their infants a hefty amount of carrots via the commercial infant food preparations. In addition, vegetarians are more prone to acquire carotenemia than non-vegetarians. The condition can also be associated or enhanced with the ingestion of carotene-rich nutritional supplements. The disease condition, including anorexia nervosa, diabetes mellitus, hepatic disorders, hypothyroidism, and renal disease, may also bestow the rise to carotenemia. Usually, the inborn errors of metabolism have more examples of autosomal recessive, but there is also a condition of autosomal dominant, like lipomatosis, in which multiple distinct encapsulated lipomas are present on the body due to the genetic defect in HMG1C gene encoding the high mobility group protein isoform 1C. So students, in this module, we have learnt and discussed about the inborn errors of peroxisomes, how the cholesterol metabolism disorders takes place, as well as a very unique carotenemia condition and lipodystrophy. These conditions are actually rare and needs to be taken care of before it turns into an havoc. Thank you.